When most people think of surimi, they think of crab sticks, the familiar seafood product found in sushi rolls, salads, or frozen foods. But scientifically, surimi is not a finished seafood product at all. It is actually a highly engineered protein system made from fish muscle, designed to create a specific texture through controlled processing. Surimi can be described as a concentrated preparation of myofibrillar proteins, the structural proteins responsible for muscle function in fish. These proteins, especially myosin and actin, are the key to surimi's unique behavior. Under the right conditions, they can unfold, interact, and form a three-dimensional gel network that traps water and creates the elastic, cohesive texture that defines surimi-based products. So in essence, surimi is not simply processed fish. It is a carefully designed protein material whose structure and texture are built through food engineering. Surimi production begins not with a finished seafood product, but with fish muscle selected for its functional protein properties. White muscle is especially important because it contains high concentrations of myofibrillar proteins such as myosin and actin. These proteins are responsible for forming elastic gel networks during processing. Raw material freshness, low lipid content, and limited protein degradation directly influence the final texture and quality of surimi. The process begins with the selection of raw fish. Not all fish are suitable for surimi production. Manufacturers usually choose lean, white-fleshed fish, such as Alaska pollock, threadfin bream, or other species with low fat content and high concentrations of functional myofibrillar proteins. This is important because fat interferes with gel formation and oxidation stability, while dark muscle pigments can negatively affect color and flavor. Freshness is also critical. If fish proteins begin to degrade because of enzymatic activity or poor storage, their ability to form strong gels decreases significantly. Since texture is one of the most important quality attributes in surimi, the quality of the raw material directly determines the quality of the final product. After the fish is cleaned and mechanically deboned, the muscle tissue is minced into a fine paste under carefully controlled cold conditions, typically around 5 to 10 degrees Celsius, or sometimes even lower in modern processing plants. Temperature control is essential here. Proteins such as myosin are highly sensitive to heat. If the temperature rises too much during grinding, these proteins can partially denature too early, reducing their functional ability before processing even begins. Once the fish is minced, the process enters one of its most important stages, washing, sometimes called leaching. This may sound simple, but it is actually a sophisticated separation step. The minced fish is repeatedly washed with chilled water to remove water-soluble components such as sarcoplasmic proteins, blood, pigments, enzymes, low molecular weight compounds, and some lipids. What remains is a protein-rich fraction that contains mostly insoluble structural muscle proteins, especially myosin, actin, and actomyosin complexes, which are the proteins needed for gel formation. This washing step improves several things at once. First, it increases the concentration of functional proteins. Second, it removes pigments and compounds that contribute to fishy odor. And third, it creates a whiter, cleaner, more neutral base that can later be transformed into many different seafood analog products. After washing, the fish protein concentrate still contains a large amount of water, so the material is partially dewatered and refined to remove excess moisture and unwanted tissue fragments. At this stage, manufacturers usually add cryoprotectants, such as sucrose, sorbitol, or phosphates. This step is especially important because surimi is often frozen and stored before being used in further processing. Without cryoprotectants, freezing would damage the proteins. Ice crystals would disrupt the hydration layer around protein molecules, causing irreversible denaturation and aggregation. Once that happens, the proteins lose their ability to form elastic gels. Cryoprotectants help stabilize the protein structure during frozen storage by protecting molecular hydration and reducing freeze-induced damage. In other words, they preserve the functional protein system until it is ready for final product manufacturing. Now, when frozen surimi is later processed into products such as crab sticks, the next critical step is the preparation of surimi paste. Salt is added first, and this is one of the most scientifically important steps in the entire process. Salt helps solubilize myofibrillar proteins, especially myosin, allowing them to move out of their tightly packed muscle filament structure and disperse into a sticky protein matrix. This creates a viscous protein sol that will later transform into a gel when heated. Without salt extraction, the protein network would not develop properly. Additional ingredients are then incorporated depending on the final product. Starch may be added to improve water binding and contribute to body and firmness. Egg white proteins may help reinforce gel strength. Flavor enhancers and aroma compounds are used to imitate the sensory profile of crab or shellfish. colorants create the characteristic red-orange surface associated with crab stick products. At this point, the mixture is no longer simply fish protein. It has become a formulated protein composite designed for both texture and sensory appeal. The next stage is shaping. For crab stick production, the paste is often formed into thin sheets rather than simply molded into blocks. These sheets are layered, aligned, and folded to create an anisotropic structure, meaning the material develops directional fibers that resemble the muscle strands of real crab meat. This structural engineering is one reason crab sticks can be peeled apart into strips rather than behaving like a uniform gel. Some systems may also use shearing or extrusion-like forming technologies to improve fiber alignment, but the goal is always the same, to mimic the fibrous texture of natural seafood muscle. Finally, the shaped product undergoes thermal gelation, the most critical transformation in surimi science. This heating process is usually carried out in two stages. The first stage occurs at approximately 40 to 50 degrees Celsius in a process known in Japanese surimi technology as suwari, or setting. At this temperature, myosin molecules begin to interact and form a preliminary network. Protein chains partially unfold and start linking together in an organized way. This creates the foundation of the gel structure. The second stage occurs at higher temperatures, typically around 80 to 90 degrees Celsius. Here, proteins undergo more complete thermal denaturation. Hydrophobic regions become exposed, molecular interactions intensify, and a permanent three-dimensional gel network forms. Hydrogen bonds, ionic interactions, hydrophobic interactions, and in some systems covalent cross-linking all contribute to the final structure. What forms is not simply cooked protein, it is a protein-water composite gel, a microscopic network that traps water inside its structure while maintaining strength and elasticity. This explains why high-quality surimi products are not just firm, but also juicy, springy, cohesive, and able to tear into fibers. In food engineering terms, surimi production is a remarkable example of structure design through process control. Every step, from raw fish selection, washing, cryoprotection, salt extraction, formulation, shaping, and thermogelation, influences protein functionality at the molecular level. And those molecular changes determine microstructure. Microstructure determines texture. And texture determines how consumers perceive quality. So, although a crab stick may look simple on the outside, what it represents is actually a highly sophisticated process of protein engineering, transforming fish muscle into a carefully structured food material through science, chemistry, and controlled processing.